Hello.
Hello, Beth. Good to see you.
How are you?
Peachy keen. How about yourself?
Doing great. Doing great because we have yet another viewer question.
Woohoo!
Mhm. So we have another question from Kavya from Helsinki, who wanted to know, I've been trying out feature transformations for cell painting data. I see that even Broad publications have multiple methods. How do you decide on this and also automate it so that any new features you measure in the future can also be transformed to have a Gaussian distribution?
Whew. Love
Kavya's
it.
pulling out the hard ones, honestly.
Yeah, I'm glad you took this one.
Yeah. Um, this is where my catchphrase comes out, which is the it depends. And unfortunately, I wish we had a really good answer for why features get treated in certain ways, but this is something that's an active research area. It's also something that's different for different kinds of features. And so I've pulled together a couple papers to just briefly highlight some important aspects here, but I'm not really going to answer your question, Kavya. I'm sorry. Other than we try to find a best practice and then we stick with it, but then every so often we reevaluate our best practices. So this is from the Caicedo et al. Nature Methods 2017 paper, which is a very popular, well-cited paper, and is the paper that sort of was based on the first meeting of the Cytodata Society. And basically, actually, almost everybody in the world who was working on image-based profiling at the time, like, got together and wrote this paper of like, what are we doing? Like, there's this new field of image-based profiling. Like, what are we doing? And there's a lot of really great stuff in here about what are the sort of workflows that people do, but specifically here, one of the things that was pointed out is different features actually can behave super differently and can have very different data shapes.
Mm.
So things like this particular texture measurement looks a pretty normal distribution, and I say that like in the mathematical definition of normal. Um, Whereas this one is normal-ish but right shifted. Some of these are bimodal, some of these are, very, it's something where it's a particular number, and so there's a finite set of values that it can be. There's not a continuous distribution. Here's, a beautiful intensity histogram that will look very familiar to any biologist who's ever done FACS or anything like that, where we can see we have a G1 population and a G2 population. So all of these things are different. And there often aren't good ways to necessarily say, how are we going to transform all of these? So what are the common things? So one of the most common things is just, well, we say, oh, we're going to take the median and the MAD and we're going to normalize these measurements. And yes, in some of the cases, the median is going to not be where the mode is, but that's just something that we deal with. And the fact that we take so many features and that these features do have these distributions, again, I come back to like, here are two different texture measurements that have different distributions. We can then say, well, by looking at all of these and by normalizing them with the median to sort of at least make sure they're in the same scale, since some of these go from negative to positive, some of these are minus 100 to plus 100, some of these are 0 to 1, we can at least get a sense of how far we are away from, you know, the middle of the response, whatever that is.
Mhm.
Um, Obviously, median isn't perfect here, and you're not going to get a Gaussian just by doing a median normalization, but you will therefore have things that respond differently, and you will therefore have lots of measurements that are contributing. Um, that's kind of an unsatisfying answer. Um, Other things that folks at IPRA and have done in the past, so this is from Robon et al., where they said, well, we have cases where the sort of mean and the standard deviation are the same, and so like the individual feature distributions um, will cover the same thing, but maybe one is, very sort of uh, full distribution, and some of them are very sort of bimodal in population. So let's just throw out trying to sort of look at these things together at all, but instead we will say, let's look at how the medians change between all of our samples and say which perturbations cause changes in median, which perturbations cause changes in MAD, and try and draw connections between
Hmm.
samples in all of these different spaces, all of which will behave slightly differently, and then say we'll use, in this case, a technique called similarity network fusion to say, and then what we'll look for are connections that persist over time. So, like this connection here persists across multiple interpretations of the data and different ways of shaping the data. And so, therefore, this is probably a stronger connection than, a connection that you only see in some ways of looking at the data, but not others. Uh,
So
other,
so doing that
Mhm,
rather than so in the very first example, you were basically saying, okay, we're condensing the information for this single feature, in a single
mhm,
feature to its remaining one feature. We're picking, median, right? But
mhm,
here you're actually increasing the number of features because each feature you're finding the median of that
mhm,
value, the MAD of that value, and so you're actually
mhm.
expanding the number of features that you have when you're
Yeah,
taking this
and
approach.
looking at
Okay.
the data from essentially multiple angles.
Hmm.
Um, this is a recent paper from Alex Liu's team at Microsoft, and what they said is, in their case, PCA, helped a lot by just saying, okay, let's forget about the individual features for the purposes of making clusters, because really what we care about is the clustering and not so much the individual features. And so we will do principal component analysis, which will allow us to sort of say these sets of features all report on similar things.
Hmm.
And rather than sort of worrying about the distribution of any of them, we will just sort of say, well, let's look at principal component analysis. And they did this for a bunch of different kinds of features. And what they said in the paper is for CellProfiler features, which can have these sort of like very crazy distributions, PCA helped a lot more than these ones that are deep learning based, and so that therefore are um, usually starting in a normal distribution because deep learning features are often in a normal distribution. And so we might do transformations on our features. So PCA is one way of doing it. This is from the Arevalo et al. Batch correction paper. And there they just said, let's try a bunch of different batch correction methods. We will do feature selection, but then we will either essentially do nothing to the features. We will do sphering on the features, where we try to sort of, remove some of the variance, or we'll do a lot of different transformations, some of which are sort of reversible things, that are algorithmic. Some of them are deep learning transformations, and we will see how well we can sort of keep our biological knowledge, and remove our batch correction knowledge with all of these different kinds of transformations. And what they showed is no one thing was better than the other, unfortunately. Um, and we also did we previously talked about this particular paper from Asif Zaritsky's group, but one option also would be to, rather than saying, let's keep the original feature distribution at all, let's say, can we predict how the feature would change? And then do these anomaly profiles, and that will possibly allow us to have a distribution that is easier for us to deal with or is easier to sort of integrate. Um, I will say there are lots of space transcriptomics also deals with things like this because, gene counts can be very unequally distributed, and so there are libraries, like MOFA, that sort of have methods in there to sort of account for things like underlying distributions have different shapes. Um, but the field, and both the field of image-based profiling and the field of bioinformatics has really not settled on exactly the one best way to handle it because you might, how much you care that the shapes of the distributions are different might be really different in different scenarios. And so the answer to how do you pick which one is a little bit vibes. You look to see when people have evaluated lots of other things. So here they evaluated some things and, they say what works best. And here we've evaluated things and see what works best and come up with the best practice for you, you know, based on data, not just based on vibes, and then go from there and reevaluate when new methods come out to see if it does something better to your data. But this is far from a settled question, and how we bring together features that have all these different shapes is just mathematically hard because they all contribute something. And we don't always know what's most important for a particular piece of biology.
Yeah. Yeah, I think that's great. And I think uh we've certainly see, even in those, right, with some, like I helped on the Zuritsky paper, we saw that that method worked well, particularly well on some data sets, but not others, and wouldn't have necessarily predicted that in advance. And maybe it had to do with the number of negative controls. There's lots of ways, post hoc to guess
Mhm.
why it worked better or worse on some data sets. But sometimes you just can't know until you've tried it.
Yeah. So that's my non-answer. I'm really sorry that for both of your questions, Kavya, my answers are a little bit non-answers, but it's because you're asking great hard questions, like right at the limits of what are the things that we know, and sometimes we're just operating at the limit of things that we know.
Yeah. Well, thank you so much for asking the questions, Kavya, and thank you,
Hmm.
Beth, for leading the charge to answer them.
Yeah. Thank you for joining me. Talk to
My
you later.
pleasure. Talk to you later. Bye.